Okay, so welcome back to this next video in which we are discussing the epidermis. Okay, so we're currently discussing the stratum granulosum, and specifically we're discussing the tight junctions between the cells of the stratum granulosum, which are the things which seal the gaps between neighbouring cells and thereby ensure that water molecules cannot move across this layer. This is the layer that is responsible for the waterproofing of the epidermis. Okay, right, so we're discussing the cell adhesion molecules which are responsible for producing these tight junctions. Okay, and we've discussed that one of the major types of cell adhesion molecule involved in the formation of tight junctions are the claudin proteins, of which there are 27 different claudin proteins. Okay, well, there are 27 different genes for claudin proteins. There are, in fact, different splice variants of claudin 10, which are claudin 10A and claudin 10B, and also there are two different splice variants of claudin 18, claudin 18.1 and claudin 18.2, which means that overall there are 29 different claudin proteins. Now, they're not all found in the stratum granulosum. You know, tight junctions are found all over the place in the body. Different tissues will use different claudins in their tight junctions. The main one that's found within the tight junctions in the stratum granulosum of the epidermis is claudin 1. Okay, we then discussed that claudin proteins bind together via their extracellular loops, okay, and then they bind to the actin filaments of the cytoskeleton by binding to zonular occludins proteins, of which there are 3 different types, which then bind to the actin filaments. Okay, so remember, actin filaments are these right-handed alpha helices where you have 2 actin polymers binding, and they're both sort of wrapping around each other in these right-handed alpha helices. Okay, so if I colour one of these in pink, this one's coming out of the page towards you, it's then going back into the page like so. Okay, then this one at the moment is at the back, and then it's wrapping round out and over like so. Okay, so here is our actin filament, and basically the zonular occludins proteins are capable of binding to actin filaments and thereby attaching the claudin proteins onto the cytoskeleton of the cells. Okay, so let's now talk about different proteins involved in the tight junction. So the next type of cell adhesion molecule that I'd like to discuss is occludin. Okay, now there is only one occludin protein, okay, so this is a cell adhesion molecule, but it's not a family, it's just one protein. Okay, now, occludin has a similar membrane-spanning topology to claudins, okay, again, here is its amino terminus, here, then here is its carboxylic acid terminus, and they're both on the cytoplasmic side, and then it's the extracellular loops then that interact with one another, okay, and hold the 2 occludin proteins together. Okay, so here is the amino terminus of this second occludin, and here's the carboxylic acid terminus of this second occludin, sitting in its cell membrane there. Okay, so very similar to the claudins, and again, the mechanism by which they actually attach to the actin cytoskeleton is identical. Okay, the C terminus binds to a zonular occludins protein, okay, and then the zonular occludins protein then binds to the actin cytoskeleton, okay. So I'll colour in the zonular occludins protein again in vivid purple, and then it will be attaching to the actin cytoskeleton just like up here. Okay, so remember, actin filaments, once again, just to reiterate this, they are 2 actin polymers intertwined together in this right-handed alpha helical fashion, okay, like so. So we'll colour this one in a different colour. So here, this is looping out of the page towards us, it's then looping back into the page away from us, okay, and then the other strand is looping round towards us here. Okay, there we go. Right, so there's our actin filament, and there's our occludin protein attached to the actin filament, and these are also present within these tight junctions and help to seal up the gap between these 2 cells. Okay, so just to stress this, you have got cell adhesion molecule after cell adhesion molecule after cell adhesion molecule after cell adhesion molecule interacting with cell adhesion molecule after cell adhesion molecule after cell adhesion molecule after cell adhesion molecule on this second cell. Okay, this is how you're filling in this gap between the two cells. It's just a layer of cell adhesion molecules interacting with one another, basically. Okay, right. Now there is one final family of cell adhesion molecules which is going to be involved in sealing the gap within tight junctions. Okay, so I'd just like to mention this family. Okay, so this final family of cell adhesion molecules is the family of JAMs. Okay, and the JAMs stands for the junctional adhesion molecules. So the J is for junctional, the A is for adhesion, and then the M is for molecules. Okay, now there are actually 6 different junctional adhesion molecules. There is the junctional adhesion molecule A, there is the junctional adhesion molecule B, there is the junctional adhesion molecule C, there is the junctional adhesion molecule 4, and then there are 2 junctional adhesion molecules that are given completely different names. Okay, so the next one is called the Coxsackie and adenovirus receptor. Okay, but it is a junctional adhesion molecule. Okay, it was just discovered with regards to the Coxsackie and adenoviruses. Okay. And then the other one, the final 6th junctional adhesion molecule, is known as the endothelial cell selective adhesion molecule. Okay, so this is called the endothelial cell selective. Okay, and then it's adhesion molecule. Okay, right. So these are the 6 members of this family of junctional adhesion molecules, and these are also involved in tight junctions. So let me just discuss the structure of these junctional adhesion molecules. So basically, the junctional adhesion molecules, once again, they are integral membrane proteins which are single pass. Okay, so they have a single membrane spanning alpha helix here. They have their amino terminus extracellularly and their C terminus intracellularly. And it's the amino termini which then interact with each other. Okay, so here's another junctional adhesion molecule on the surface of another cell here. Here's the amino terminus, here's the C terminus, and these 2 extracellular domains are interacting here. Okay, so let's colour in these 2 junctional adhesion molecules in vivid purple here. Then, the way that the junctional adhesion molecules bind to the cytoskeleton is once again through binding to zonular occludins proteins. So zonular occludins proteins bind to the C terminus of the junctional adhesion molecules. Okay, and I'll colour in these zonular occludins proteins in turquoise. And these then bind to the actin filaments of the cytoskeleton, and hence that's how we form a viable adhesion between these 2 cells. Okay, so to review, in these tight junctions you have a huge number of these cell adhesion molecules which are interacting with cell adhesion molecules on other cells, and this forms an entire layer of cell adhesion molecules that completely blocks up these gaps in between cells, and this structure is known as a tight junction in between these 2 cells, and this is what you have between the cells in the stratum granulosum here, and that's how that layer becomes waterproof. Okay, right. Now, the layers then that are above the stratum granulosum. Okay, you have multiple layers above the stratum granulosum, but they're all referred to as the one final layer of the skin, or the epidermis rather. Okay, so this final layer of the epidermis that is the most outer layer is known as the stratum corneum. Okay, the corny layer. Okay, and it consists of multiple layers of cells which are known as squames, okay, or scales. Okay, now these cells have lost all of their intracellular organelles. Okay, they have no nuclei. Okay, they're not alone in that fact on my picture at least, but all of these cells prior to this outermost layer have nuclei. okay, and other intracellular organelles. The outermost layer, the stratum corneum, which is multiple layers of cells, these cells have no intracellular organelles anymore. Okay, they are absolutely full of keratin. They have synthesized a huge amount of keratin, okay, and this keratin gives them huge rigidity, so they're very, very solid, okay, and that's why they're called scales, basically, because they're not really gooey cells anymore. Instead, they are just bags of this strong intermediate filament, keratin. Okay, and these are the cells which are going to be shed off the epidermis now. Okay, so these cells are often known as squames, which is just another word for scales, basically. Okay, so this is the outermost layer of the epidermis, and its role is just to protect the under layers of the epidermis. So it's the outermost layer that we can actually see, and its role is in protection. Okay, now, gradually these squames will be being shed, okay, so that's why it's important that we're continuously making new cells. So, to review the whole differentiation process, starting with a transit amplifying cell rather than the epidermal stem cell. The transit amplifying cell will part from the basement membrane, okay, it will move up into the stratum spinosum here. When it does that, it can no longer divide anymore. Okay, in the stratum spinosum it starts synthesizing more keratin, more desmosomal cadherins, and it forms these very apparent desmosomes. Okay, gradually it moves further up. Okay, it gets into the stratum granulosum here and starts producing very tight junctions, basically. It produces lots of these cell adhesion molecules involved in formation of tight junctions, and therefore contributes to this waterproof layer here temporarily. Then the cell will move up from the stratum granulosum into the stratum corneum. It will synthesize a huge number of keratin, its intracellular organelles will degrade, and it's now a squame. Then it will be shed off from the top. Now that whole process of from leaving the basal layer to being shed off takes about a week. Okay, so it's about 1 week that it takes for the cell to completely go for its differentiation pathway to become a squame. Okay, and it goes through all of these intermediate stages on the way. Okay, so that now concludes our discussion of the epidermis.